The overall goal of the following experiment is to assess the effect of the protein of interest on neuronal migration and morphology. This is achieved by electroporating the cerebella of P4 rat pups. As a second step, immunohistochemistry of sections from GFP-positive cerebella is performed, which allows the visualization of the electroporated neurons. Next, the immunostained sections are imaged in order to measure the distance of neuronal migration and the length of neuronal processes. Results obtained will show the effect of the protein of interest on neuronal migration and morphology in the developing cerebellar cortex.
The main advantage of this technique over existing techniques, like in utero aspiration, is that the postnatal cerebellum is easily accessible.
This method can help answer key questions in the neurodevelopmental field, as it allows to unravel the mechanisms underlying neuronal migration and morphogenesis.
Visual demonstration of this technique is critical, as the reliable targeting of the injection site requires practice.
Prior to showing the in vivo electroporation of the cerebellum, we will first demonstrate a morphological assay that uses cultured cerebellar granule neurons as a model system. Begin this procedure by collecting the conditioned media and subjecting the neurons to starvation in DMEM, then mix DNA solution and calcium chloride. Add 2x HBSS and gently mix the solution by making air bubbles. Then, add 40 microliters of DNA precipitate.